Hello students. In this video, we will learn about simple permanent tissues. The cells of permanent tissue are formed by the differentiation of meristematic cells. Differentiation is the process by which the cells change their shape, size and function. Differentiation of meristematic cells results in the development of various types of permanent tissues. Permanent tissues are made of cells that have lost their ability to divide. Permanent tissues can be broadly classified into two types: simple permanent tissue and complex permanent tissue. Simple permanent tissues are those which are made of similar types of cells and are involved in functions like protection and support. Complex permanent tissues are those which are made of dissimilar types of cells that are involved in functions like conduction. To discuss simple permanent tissues, these are further classified into protective and supportive tissues. Let us first understand three types of supportive tissues: parenchyma, collenchyma, and sclerenchyma. Parenchyma is the most abundant plant tissue, made of unspecialized, living cells with thin cell walls. Structurally, the parenchyma cells can be oval or circular, or polygonal in shape. The cells are loosely arranged with intercellular spaces. Functionally, the parenchyma tissue serves to provide protection and stores food. Protective parenchyma cells are with thick walls and are found in gymnosperms. Parenchyma tissue that serves to store food, starch, latex is called storage parenchyma. For example, in potatoes, starch is stored in parenchyma tissue. Certain specialized parenchyma tissues, like chlorenchyma and erenchyma, are also seen. Talking about chlorenchyma, it is photosynthetic in nature and is seen in guard cells of stomata. Another type of special parenchyma tissue is erenchyma, which is seen in hydrophytes. Erenchyma cells have large air cavities that provide buoyancy to the aquatic plants. These are the details of parenchyma tissue. Let us now discuss collenchyma. Collenchyma is a tissue made up of elongated living cells that are irregularly thickened at the corners of the cell walls. Collenchyma tissue serves to provide mechanical support to the plant body. During heavy winds, the stems of tall plant bend, but do not break usually. This flexibility is provided by collenchyma tissue. The cells are arranged closely with little intercellular spaces. Based on the pattern of thickenings on the cell wall, the collenchyma is of four types: angular collenchyma, tangential collenchyma, annular collenchyma, and lacunar collenchyma. The last type of simple permanent tissue is sclerenchyma tissue. The cells of this tissue are long and are dead due to the deposition of lignin on the cell wall. Sclerenchyma serves to provide mechanical strength to the plant parts. It makes the plant parts hard and stiff. Depending on the form, structure, origin, and development variation, sclerenchyma cells may be either fibers or sclerids. The fibers are thick-walled, elongated, and pointed cells, generally occurring in groups in various parts of the plant. The sclerids are spherical, oval, or cylindrical, highly thickened dead cells with very narrow cavities. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear, and sapota, seed coats of legumes, and leaves of tea. Let us understand protective tissues, which are primarily protective in function. They include epidermis and cork. The epidermis is the outermost layer, that is 1 cell thick, and present in all the parts of the plant, such as roots, stems, leaves, and flowers. The epidermis is covered by a thick waxy layer called a cuticle secreted by epidermal cells. Epidermal cells have thin walls and are intervened by small pores called stomata. Stomata are responsible for gaseous exchange. The second protective layer is a cork that is seen in older roots and stems. Cork is made up of dead cells which have thick walls deposited with a waxy substance called suberin. Cells in the cork have no intercellular spaces. These are the details of simple permanent tissues. Summary. Permanent tissue is formed by the differentiation of meristematic cells. The cells of permanent tissue do not have the ability to divide. Permanent tissue is of two types, simple permanent tissue and complex permanent tissue. Simple permanent tissue is made of similar types of cells. Simple permanent tissue is of three types, parenchyma, collenchyma, and sclerenchyma.